Thank you very much, Martin. Good morning, everyone. I'll start by saying I admire you all for being 7 a.m. up, because for me, I'm scientist, I'm still sleeping. So let's keep it somewhat informal. You will have some questions as I go along. Just raise your hand, we'll address them right away, because things may just keep piling up. So the subject of my talk is kind of self-explanatory from the title, because we have three keywords there: is computers, drugs, and prostate cancer. And so basically, I will just give you a very general overview how these three work together. Main message is what we're trying to do with what I call digitized biology, because everything gets digitized these days, and that's what takes rotary phone and makes an iPhone out of it. So the only difference is really computational technologies, IT technologies, but productivity, you know the difference. So same idea is that if we start bringing computers into research, into drug discovery, that will help. And I was involved in this branch of research from the pretty much very beginning. So I started doing it very, very long time ago because I had background, like Martin pointed out, in both chemistry and computer science. Well, back then it was called mathematics. And it grew somewhat in its independent field, and it worked. So I will start off by just disclosing my conflict of interest, which I have to do. And then I'm not going to spend too much time talking about prostate cancer, because that's obviously the audience which can teach me about prostate cancer. Just want to mention 1 out of 7 will likely to have it. And what I'm going to start focusing on, that prostate cancer, unlike many other conditions, is what I would call monotargeted disease. So there is really one antichrist of everything, is androgen receptor in the center of it. Again, I don't mean to downplay many other targets, but up until now, all anti-prostate cancer, more or less, research more or less was focused on AR, androgen receptor. And this is a transcription factor, which is a hormone dependent. And again, just very briefly, upon binding to androgen, typically DHT, androgen receptor gets activated, chaperones go away, it dimerizes, goes into nucleus, attaches to androgen response elements, and starts activating AR-regulated genes. So basically, this is a general schematics. So all current anti-prostate cancer, anti-androgen therapies are built around interfering with DHT binding. So all existing drugs, and there are a few on the screen, starting with good old nilutamate, and then old falutamate, Casodex, and the latest Enza, and upcoming REM509, which is, I guess, apalutamate. They all do the same thing. So they do interfere with DHT to bind to the ligand binding site of androgen receptor. So these are competing antagonists. And again, this is a big market. So enzalutamide itself is pushing like $2 billion a year sales in the market, and REM is coming up. So you would think that the field is well covered. You would think that AR is well attended, and these drugs work. Problem is they don't. Most. So I'll tell you more specifically why. And when you look at these structures, which seem very different, but if you look from the chemist side, you'll see it's the same thing. So I highlighted in red the active side of this molecule, what I call a warhead, which actually attaches to the protein and does the business. So if you look at all of this, starting with all nilutamide, like all castration chemical, it wasn't even a drug, it's a castrating chemical. From 40s, I guess, 50s of last century, to this enzalutamide, which was bought by $1 billion cash. This was bought by $1 billion cash, too. And now it's being pushed. It's the same thing. So the same red warhead. So this is, I can talk chemistry if you like, just to give you some flavor. Aromatic ring with 2 electron withdrawing groups side by side in ortho position, and a peptide bond. Yeah, I have to, sorry. Thank you for reminding me. So this is, right. So this is essentially, you see these substructures, this is the same thing. And here is just drawn different, but this is the same CH and CF3, CN and CF3, CN and CF3. Actually, there's a mistake, that's okay. And a peptide, it's the same thing. So in principle, all the reasons by which the new drugs been developed, and the reasons are because the old drugs fail and prostate cancer goes into CRPC, they're not going to be changed. It's all the same chemicals, same mode of action, same everything, just different dressing. Better pharmacokinetics, little bit less of side effects, but essence is the same. So no wonder prostate cancer gets into CRPC stage driven by mutations on the ligand binding side. And the ligand binding side, that's where those guys bind. That's where all of those molecules bind. So AR is everything in life. On one hand, this is monotargeted disease. So you take care of that protein, prostate cancer is suppressed. Flip side is apparently that this is such a smart and mobile and plastic protein, unlike anything else. Like honestly, I tried to find any example when the protein drug target would mutate one amino acid and switch drug into agonist. I didn't see any other examples. I'll be happy if you point me something I don't know. So AR is so central, so paramountly important target that it can do a lot of things. So what it does, it mutates one amino acid at ligand binding site, and it can mutate different ones. And all of a sudden, Casodex becomes agonist, flutamide becomes agonist, Enza becomes agonist. Meaning, you give more drug, you get more cancer. Because all of a sudden, protein starts feeding on the drug. So drug becomes acting activator, like GHT itself. And so that's one of the reasons, not the only one, but one of the main factors why all of a sudden prostate cancer relapses into CRPC. Because that's already mutated AR. And just stepping away a little bit, we started looking deeper into this mutation scenario. And we employed here at Vancouver Prostate Center cell-free DNA technology. So basically, when tumor shreds a little bit of free DNA into serum, into blood, we can detect that using deep sequencing technology. So we can start monitoring what kind of mutations appear in AR, among other proteins. And we so far, including literature data, know about 30 plus or 40 mutations which occur in the ligand binding domain of androgen receptor. All occur on exon 8 here, and you see there are all kinds of them. So basically, AR can mutate on the ligand binding site whichever way it likes. And that gives enormous plasticity to this target. Again, it can react on any kind of drug in a way that the drug all of a sudden becomes agonist. And here with stars are marked those mutations which have been documented in the literature that they actually do some sort of agonization. So, and mutations are all over the exon 8. So you can see that they span anywhere from beginning to the end, and moreover, there are more mutations occurring on the more like N-terminus, so anywhere from 700 position to 920 position, 919. So you would think that all span of this exon is in fact can be mutated. And that's where we, little by little, start showing you how computer modeling can start bringing some rationale into things. The simplest thing we've done, we actually mapped all these mutations in a three-dimensional structure of a protein, of AR, and all of a sudden you realize that these mutations are not all over the map, not all over this exon, but actually all pretty much around the ligand binding site, all around the testosterone or the dihydrotestosterone binding area, all here. And that made me think immediately that these are all functional. I think not just those marked with star are functional, but all of these are. And so what we've done next, in fact, we made all these ARs. We made them all, we screened them all against all kinds of drugs and steroids, and guess what? Every single one of them, of these 38 mutants we studied so far, is functional. It does something. It either sensitizes things to drugs or sensitizes things to steroid. Estrogen all of a sudden works. Or some of the mutants, and I'm not going to go into too many details, but this is all published in this genome biology paper, if you're interested in following up. So some of these, for instance, make AR more sensitive to DHT 6-folds, 4-folds. So it takes DHT in, but it takes just a fraction of what it used to take. And here's one example how these mutations activating drugs into agonists are very critical. One take-home message, of course, is that every patient, in principle, has to be monitored for these things, every CRPC patient. Because you can see these mutations popping up, it tells you you've got to change the regimen. If someone is on Casodex, like here, and you see these mutations, so this is an actual example, a patient was sequenced for AR mutants, and two dominant mutations have been observed. This 889 and 896, this is a classical Casodex activating one. And here you see that scenario I told you. More drug you give and they go from 0 micromolar to 50, more PSA up. more activation of prostate cancer. And the concentrations of this drug in plasma are very high, probably around 20 here. So you get quite a bit of activation. You see up to 300% of AR up there. So clearly this patient had to be taken off Casodex because two dominant mutants, let's call them red for the sake of illustration, are present. So the patient was switched to Enza. 4 months later, guess what? These two mutants shrunk to about 20%. And there are 80% new mutations occur, which now resistant to Enza. Same story, more Enza you give, more progression of prostate cancer you have, more PSA is up. And all of these 4 mutants in different extent, but all of them get activation here at the higher concentration. And keep in mind that Enza can be as high as 70, 90 nanomolar in plasma in the tumor. So these 50 micromolar, not to mention 2 or 3 here, definitely are there. So this mutant will be activated. So that's a very illustrative example how the current therapies start failing. Even though the Enza is on the market, the RAN509 is coming, same thing. And guess what? RAN is doing the exact same thing. Like there is no difference between Enza and RAN in terms of activating the mutants. To make situation worse, as we all know, that CRPC really shows significant presence of truncated forms of AR. Here are a few dominant truncated AR forms where here's the full length you see, and this ligand binding domain is entirely missing. Specifically, ARV7 is the one which is most present. ARV567 and a few others are also documented. So basically, the entire LBD is missing. And ligand binding domain is that where all the drugs bind. So all these splice variants, and keep in mind they're actually constitutively active. They don't need drug, they don't need steroid, they don't need anything. They just all fully on in CRPC. And clearly no current drugs address this at all. Because again, all of those guys bind here, not to mention that again, mutations occur in the LBD. So that really keeps AR on top of the game. That's why it's very hard to come up with meaningful therapy for this target, because all the time it finds a way. And I'm not aware of a same plastic, same sophisticated target. And keep in mind, probably it's the most studied drug target of all ever in my records. So we decided to exercise completely different approach. What we decided to do, we decided to look into different ways of targeting this beast. Rather than going again after LBD with another new analog of Enza, but many companies do that, and we know that. And now the Bayer is in the game, and many others, with all this new ODM201 and other compounds. Aranda is doing it in Finland. They all still go after this LBD. I don't know why, maybe because the market is still so big, but they keep doing it because probably you're going to make you $1 billion no matter what. So what we decided to do, we go after what's conserved in all these proteins, what's conserved in full length, what's conserved in splice variants, and what is conserved. Actually the DNA binding domain, the green one, the DBD. And if you think about it, this is the active site of a receptor. And that used to be, it is still a big misconception in a way that ligand binding domain of androgen receptor, in fact, any nuclear receptor, take estrogen, progesterone, glucocorticoid, that's where the steroids bind, that's the active site. It's not. The active site is DNA binding domain because these are transcription factors, they transcribe proteins. That's the business end of it. It sits on the DNA with DBD, starts reading, that's the action. The steroid binding site is a switch, it's an allosteric site. It's an on-off thing, it's a regulation, it's not the active site. There is nothing active about it. So, and that's part of a problem. You hit something which is not essential, like LBD, well, protein can kind of tolerate the hit, can try to evolve a few other things, because this is not active site. It's not really that dependent on this. And that's why it can play all the evolutionary games. What it is dependent on, a DNA binding domain, the reading head, that's where we should hit. And that's what I said we're going to be doing. We're going to hit here in the green, in the DNA binding domain, and if we are successful, we're going to kill them all in one hit. Like it's all these birds will be killed with one stone, no problem. What is the problem? Of course, problem is that DNA binding domain is an active site, meaning it's conserved. So it's such an evolutionary engaged part of a protein, the most important one, that in fact all transcription factors use it. So the level of conservation between AR, ER, GR, and PR in that particular area is like 95%, 9%. And that's why no one ever approached DNA binding domain as a drug target, because the conventional outlook is that you target something so conserved, your off-target effects will be immediate, and you will hit all these 4 proteins in the same time. So what made us still do it? Computers, so the computational technologies, because what I've noticed is there is still 2-3 amino acid differences here around 592, 594 areas, which I thought will be enough for me to develop something selective. Because I don't screen drugs from wet lab panels. What I do rational design. So I thought I will design compounds so rationally that I will engage this exact residue. And that's where the difference between AR and others will play in. So fortunately, the crystal structure of that DBD, that DNA binding domain, the green part of a sequence, was available. So here it is. AR sits as a dimer. So here is a sort of a orange and green sitting on the DNA as a functional dimer, as that reading head. This is the active site. And it was resolved. So we took that structure, put it on a computer in 3D simulation, looked at every possible angle of this dimer, took it apart. You can do it on a computer, and examine every possible surface, every atom, every residue using computational algorithms. We draw the surface to figure out where the cavities are, and we started figuring out how these cavities can be targeted. And we use our standard protocol, what we call virtual screening protocol. So by that time, we had about 550 million molecules on files. These are hypothetical structures, so these are not real molecules on real shelves. These are files with the structure of a compound. But what we can do in a 3D virtual reality, in computer world, we can have a 3-dimensional model of a protein, we can have a 3-dimensional model of every one of these molecules, and we can run screening protocol, which is not working here, thank you Steve Jobs, to see if compound sits well inside the active site, because that's how drugs work. It's a key in the lock principle, discovered by Emil Fischer late 19th century. So basically, drugs would sit ideally inside the cavity. These days, actually, we have 1 billion molecules to go through. The field is expanding, it's exploding so fast that when we started this project about 4 years ago, we had 50 million, now we have 1 billion. 1 billion compounds we can actually buy. These are purchasable. So anyway, we start using computers to funnel down from 50 million to 1 million, so we see compounds which sit inside more or less okay, and then we use more and more and more and more algorithms to narrow down to a few hundred compounds we can actually screen in the lab. And that's my job. So here's basically my life story in one slide. That's what I do. So I start with billion compounds, I'll give you 100 which I think will work. And they do. So when we started this, with all kind of algorithms, we've been then assaying a few hundred compounds using all kind of wet lab screens. So we of course had a standard PSA assay on androgen receptor and EGFP transcriptional assay, essentially seeing if compound suppresses the activity, the transcriptional activity of AR. If it does, then we go in a V7 assay. So when that truncated isoform is very specifically checked, then if it works, we look at the cell proliferation assay, looking all kind of prostate cancer cell lines, see if cells get suppressed. Then, of course, we do the BLI. This is a physical measurement similar to SPR. Essentially, there is a piece of equipment. You attach AR to the chip, and you wash compounds by it. If compound sticks, then the mass of a chip changes, and you see the detection in a nutshell. So this is a confirmation that it's physically, small molecule physically interacts with your target. And then, of course, we did the mutation assays. I'll go into more details. When you mutate out that particular amino acid you think you are hitting, you should see that effect is compromised. Messages here really is that it goes in many iterative circles. So we find this few hundred compounds, we screen them all, then we learn what worked, what didn't. We fine-tune the pipeline, we do it again and again and again. And every round, we become more and more and more sufficient. So here's an example. Yes, so at the end of it, because it's not part of your broad pipeline, but once you find your lead compound, the leading ones, then you go into specificity, of course, and then you go many other screens, such as you look at stability, you look at toxicity, you look if it stays in plasma, you look at if you go in vivo, look at the PK. But this is general high throughput pipeline. I've shown you when hundreds of compounds get routinely checked. Once you find your winner, yeah, you start drilling in on all kinds of things. I'd say we would at the end of the day we would look at 300 parameters for the compound, which will include a broad kinase panel, see if it's not compromised. You would see things like classical off targets like HERC. This is ion channel and heart. You really try to avoid. This is anti-Christ of drug discovery and all other things. But that's later down the pipeline. So right now I'm talking about the optimization cycle, how you start with 50 million and get something which actually works. Because obviously by chance you're not going to find it. One more number I can throw in. So traditionally without computers, people would just screen wet libraries. So big pharma, they all routinely have up to 2-3 million compounds physically on shelves. So they physically put robot and screen. So success rate of that is 0.03%. And typically what you pull out of it is either what we call dirty hitters, so compounds which hit across the board, or you would probably pull out something which is natural product-like, meaning that the chemistry around that will be very, very difficult, or there will be some kind of issues. When we use computers, we kind of can factor in things we don't want to see or we want to see in a compound. We can factor in toxicities, because we kind of more or less know what the toxicity substructures and flags are. We can factor in things like solubility, drug-like, and on and on and on. Anyway, we do it in a rational way. And versus 0.03%, when we are on target, our hit rates are about 40-50%. The lowest we ever had probably 5%, but it's still 5 versus 0.03. And again, going with computers, when you model everything, you see everything, again, it makes your life so much easier because you really can control the fate of your drug prototype. And so here's the first round results of the first screen. So when we screened first 100 molecules, we identified few which all demonstrate IC50 values for both PSA and GFP. These are equivalent assays, around 4 to 10 micromolar. You can see these numbers correlate more or less, which is another good sign. So that's typically our starting point. When something works with better than 10 micromolar, we call it a hit. So something is worth following up on. And you can see there was quite a bit of diversity of compounds. So it's not just the variety of the same thing. And so the second round, we kind of came up with a hybrid of a couple of hits. So that's when already we started making custom-made molecules. So again, this 50 million we had, these are something we can buy. Each cost like $50. And that's in fact what makes now academia a big player in drug discovery. Because with computers, with academic budgets, we can actually start doing drug discovery. Because running 2 million in big pharma with robot, the whole exercise is about $1 million US. Here, 100 molecules, you buy each about $50 to $100, 5 to 10K budget for initial screen. Very different. And so out of this initial screen, then you find some hits, then you start customizing your compound, making them better, better, and better. And here's what happened on the first round of MedChem. We took this very specific scaffold we liked, and then we started playing with rings A and B and C, changing them a little bit. And you can see IC50s went from 5 to 10 micromolar to sub-micromolar range. You see they're all below 1. This is already something. You keep pushing up until things working out. So the second round, that's where we already fixed. So previously we would play with 3 rings, A, B, and C, right? So we were happy with B and C. We fixed them. We kept playing with ring A, trying all kinds of substitutions here. And you can see our IC50s went as low as 100 nanomolar. And that's what enzalutamide has, about 90 nanomolar. So this was a compound we call 14449, which was already as active as Enza. But again, that wasn't really what we were after. Yes, it's as good as Enza. So on full-length AR, on normal AR, you wouldn't even distinguish 2 inhibition curves. So Enza is like 80 nanomolar, we are 100 to 90 nanomolar. You see two graphs not even separated on the GFP, and essentially same thing on PSA. So it did work as good as what's on the market. And keep in mind, back then, Enza wasn't even on the market. But when we go to enzalutamide resistant cell line, where that specific mutation which activates AR is present, the scenario is very, very different. So Enza starts agonizing AR and starts driving the cell growth at about 200 nanomolar, just about where it should be. And after that, after 200 nanomolar, more Enza, more cancer growth. And keep in mind again, in real system, Enza goes as high as 50 to 90 micromolar. So something like 40 times higher than this. While our drug worked fine, as expected. Here is the orange line. So what we could already see, okay, so this new mode of action, when we don't hit LBD, ligand binding domain, when we hit the active site, looks like the strategy works. So we do avoid these point mutations which cause CRPC. But again, it wasn't really what everyone was after. Again, everyone was after V7, when LBD is entirely missing. Because to date, there wasn't a single chart published when V7 was inhibited. Single one wasn't in the literature. So everyone was looking for this chart. So that's where, or the next one, I think. That's where this 14449 could inhibit V7. This is not the chart yet. These are different cell lines. Yeah, sorry. PC3 wasn't affected, so when AR is not present, no effect. So it is on AR. Again, this is a standard cap CRPC cell line, and that Enza resistant MR49F, they inhibited down to 0. And that's the binding site. That's how this 14449 sits inside the target, the GBD. So we could evaluate every interaction, every single hydrogen bond, every hydrophobic interaction, every atom. So we can dissect how this drug acts down to the atoms, down to orbitals. Again, we have a 100% rationale, why it worked, why it's selective, on and on. If you look at all those mutants we see in cell-free DNA samples from the patients, all those mutants inhibited by 14449, no problem. You don't see at the end sort of crippling up effect at no panels. Again, this is how all the ligand binding domain mutations are addressed. But again, everyone was after V7, and that was the chart. That was the chart which really solved the idea. So this is the actual transient transfaction with V7 when AR is truncated. There's no ligand binding domain. And that's where Enza stays at solid 100% activity cell line, baseline. And that's where 14449 worked. So with about IC50 of 5 micromolar, that drug could inhibit V7. Again, this is first ever published V7 inhibition curve. No one ever shown it. Some people claim they have it, never actually shown this chart. So that's where the tone was really different. That's when people kind of realized, okay, you can target DNA binding domain. Of course, as always, question is selective that action or not. Because again, this is 99% concern. So here it is, selectivity chart. It is selective. So if you look at 14449 action on PR, no effect. On GR, no effect. There is a little bit of effect on ER at higher concentration. Just to be fair, Enza does the same. Enza doesn't affect PR, GR, and these are things you don't want to touch, especially PR. Enza does hit ER, but actually it hits stronger than we do. And there is also always rationale. So here, this amino acid, the anchoring one, the selectivity amino acid, is a bit different. For ER, it's closer to, and it's closer to AR, so there's some effect. But generally, this is selective action. You can tell that GRPR stay at 100% no matter how high you go in concentration. Effect on ER is not that pronounced as for other anti-androgen and not that significant anyway. And AR is hit very, very nicely. So V7 assay worked here when the entire LBD is absent. Selectivity is respected. So the drug actually works. So the new mode of action against androgen receptor was established.
Would you call that a drug or
Drug prototype, not drug by far. And then, of course, we needed to pile up more and more evidence that, in fact, the drug is doing what it's supposed to be doing. Because the ultimate answer that the drug is doing what we say would be the crystal structure of DNA binding domain with DNA and our drug sitting inside of it. But that's unfortunately something we couldn't achieve because just resolving that available crystal structure of GBD took like 13,000 iterations, and there is only one structure published. We talked to the guy who did it. In fact, we had a grant with him. We worked with him from Dan Garrett from University of Buffalo. And out of 89 structures of AR in protein data bank, just one is this DNA binding domain. Because he his career solving it. And now when we throw small molecule in, it was too much to handle. So we cannot have that ultimate golden standard answer that drug sits inside the active site. So we had to build secondary evidence confirming the action. Again, one is V7 assay, one is selectivity assay. We had few pieces of in silico confirmation. We'll see if the movies work. So basically, no, it doesn't. Okay. Basically, what we've been doing, we would take this molecule and this part of a protein and run molecular dynamics simulation in the computer, so kind of let it move around and mingle and see if compound finds its hole in the protein and ends up there, and it does. And then we did mutation. So when we mutated out those anchoring residues, we could see that the effect of a drug starts sort of decreasing. So that tells you that these are actual amino acids which get engaged. But by the way, we're not really concerned with that in terms of real life scenario because we don't see these mutations in AR ever. And we don't think that GBD mutations can naturally occur because the function of a protein gets compromised too much. But Martin pointed out very rightly that it's not drug yet. Again, this is a small molecule which looks great, which acts the way we want it to act, very active. Again, IC50 is 200-300 nanomolar in different systems. Problem was it wasn't stable. So what we use mouse and rat and human microsomes to see how quickly compound gets processed in this artificial system. There are a whole bunch of live enzymes present, so we see how quickly this molecule gets eaten up by a body. And this is called half microsomal clearance time. So basically, this is time which takes for 50% of compound to disappear. And so this number, the higher the better. So depending on the microsome we've been using, so in mouse microsome was supposedly only 14 minutes, in human microsome was 40 minutes, so that wasn't a very good number. It means that if you put this drug inside human body, in 40 minutes half of it will be gone, or even faster. So it's not drug yet. Just like I said, once you zoom in the prototype, there are so many other things you have to start tweaking. So we started tweaking the structure, but again, it would take time. We decided to go in vivo anyway. Even though the mouse microsomal was only 14 minutes, we decided we still do some pilot study. We're just going to go very high dose, very high dose. So we went 100 mg per kilo twice a day, and out of 3 routes of administration, we went IP, because here you see that when it's IP red line, the clearance of the drug out of serum is somewhat not as rapid, even though it's tremendous. Again, you start with what, 15 micromolar, in 5 hours it's half micromolar, in 10 hours it's pretty much gone. But nonetheless, we knew that the drug is non-toxic. That's something we could control. And so we did in vivo anyway, just to establish proof of principle, of course. And we did a xenograft on M49F tumor. That's the Enza resistant ones. And the gray line is a control, is a vehicle. And the red line is when you administer Enza. So just like you would expect, the Enza promotes the tumor growth. Tumor volume goes up significantly above the control line. And the yellow line is our drug, 14449, and you can see it works. So we did see statistically significant decrease of tumor volume when the 14449 administered. Again, even though we knew this is extremely unstable drug, we've been just loading and loading and loading IP, and fortunately animals could take it, and we could see statistically significant reduction of the tumor. That's another tumor which is not resistant type, and that's where Enza works, of course. The gray line is a control, and our drug also worked. Not as good as Enza, but again, that's understandable. This was a very initial prototypical molecule. So, what could we do about making it more stable? Again, the difference between rational and computer-enabled drug discovery and just the empirical one, we can control things. As I said many times, I guess that's one of the learning objectives. We predicted what kind of metabolic processes can happen to the drug. We have software for that too. So this is the chart where software would map this particular 14449 compound to different cytochromes and would predict what kind of chemical modifications can occur in this molecule. You could see that one of the libel point was this morpholine ring. So all of a sudden hydroxyl gets attached to it, on a different position gets attached to it, or morpholine ring gets fallen apart, gets sort of opened. Here the morpholin ring gets cleaved altogether. So mostly we could see that most of the attack happens around this area, the morpholin ring. So what we've done next, we've developed some analogues where this morpholin ring was modified. Again, this is not exact structure. I couldn't show it at the time. So we started playing with this morpholin ring. And we did the next analog we called 512. That's where the microsomal clearance time went from 14 minutes to 60 minutes. And still the activity of a compound was maintained at about same 150 nanomolar range. And then we did one extra modification. We went to compound 14518, and that's where microsomal time went to 260 minutes, about 4 hours. And this is something which already could qualify the molecule for oral drug. Because the clearance is 4 hours, it means that, twice a day that could probably be good pill. And in fact, we could even push some of the activity numbers even lower. So this is at the level of sensitivity of assay. So we had about 100 nanomolar compound and 4 hours half clearance time by playing with this ring. And the reason I cannot show the real structures, you'll find out later. And more importantly, by the time, it took time. It took us about 3 years really to optimize the compound from just really to go from idea to having this working prototypical molecule, about 3 years. That's another take-home message, is that when you use computer-aided drug discovery, your lifespan of a project is very short, because computer helps you to propel through every stage. And so 3 years later, after the first moment, I said, why don't we try GBD to actually have something which is prototypically working? Well, 3 years. In these 3 years, new cell line appeared, this 22RV1, which is believed to be V7 driven. Because by the time we've been doing all kind of experiments, we didn't have this one. We use M49F, which is Enza resistant. And this is believed to be V7 everything resistant. So when we administered that latest 4-hour half clearance time compound, 14518, it worked. So with IC50, as you can see, roughly 7 to 10 micromolar, which is very acceptable. Not surprised that Enza really stayed up. And so basically, that was the ultimate answer we've been looking for. And by the time we got multiple offers to sell the drug, to license it, and we did out-license the whole project to Roche, and that's why I cannot show the structures here, because now it all belongs to Roche, and we out-licensed it for a deal which is $142 million, the biggest academia licensing deal in Canadian history. So we beat Research in Motion and everyone else. So that was a very good success story. That gives credibility to everything we do with computers. And importantly, it sets new directions. So last few slides, I want to talk to you about not this specific example, but rather how we can make it a generic thing, how we can do the same thing for other targets, how we can clone the success. So currently, really, the computer-aided drug discovery technology is very, very mature. So we can do, as you can see, we can tweak a lot of things. We can control a lot of factors. We can control metabolism, definitely target effects, selectivity. Computers work. So the real bottleneck right now is the structures. So if we wouldn't have that DNA binding domain structure of an AR sitting on DNA, we wouldn't be able to develop these drugs. So I need to know every atom. I need to know every potential hydrogen bond. In fact, I need to know every trapped water molecule. Sometimes water gets trapped inside the protein, and this is a very important bridging substance sometimes. So targets are the bottlenecks. And so that's why we put together that CFI, which Martin mentioned, to really address this bottleneck and to enhance UBC with structure resolving capabilities. So what happened in recent years, what's called the cryo-EM technology. This is basically a new way to solve structures of proteins. Traditionally, it will be NMR and X-ray, and those are notoriously expensive, difficult, and really kind of skill-depending art. While cryo-EM can become sort of high-throughput way to see molecules. So essentially, you can see molecules with super powerful microscope these days. And these are some examples when molecular structures are resolved with cryo-EM. And we have those specialists in UBC. And so what we propose to do by adding this cryo-EM, we propose to really enhance this pipeline So when you go from genomic data to individual proteins, then you resolve the structure using cryo-EM, and then we can very quickly come up with drug candidates using this computer-aided drug discovery technology I was showing you. So we decided to put this together, to max it up, to put it in a sort of generic way that you can, once you have genomic idea, you can generically generate protein, generically resolve its structure, and generically design small molecules for it. So we would develop this platform, which we call Accelerated Drug Discovery using Clinical Translation, ADDUCT, and that's what CFI awarded us with, and thank you CFI for that. So this is very Canadian enterprise with all the main components being stitched together. And the platform, of course, is sophisticated, so we have everywhere from genomics data. Again, I mentioned those cell-free DNAs, so this is an example how we can take genomic data from patients, gives us all kinds of right ideas. Then we have this protein core, because everything has to be going through proteins. And then there is a structural core in the center of it, using cryo-EM, we solve three-dimensional structure. We understand molecular basis for the function of a protein. And the CAT platform allows us to match protein to small molecule. And then, of course, there's down the line optimization things I told you, what called admit absorption, distribution, metabolism, excretion, and toxicity. Make compounds more stable, make them less toxic, make them stick to the body, and then, of course, the clinical trials. So our vision is that we will have all these critical components of drug discovery pipeline here in Vancouver Prostate Center in UBC, structural biology, genomics, clinical trials, diagnostics, screening, optimization, and what I lead, the computer-aided drug discovery. So that pretty much rounds up my talk. I have to acknowledge a lot of people who were involved and a lot of granting agencies, and there is that picture of us after the licensing deal. But essentially, this is the main chart. That's how we stitch it all together. And at the core of it, essentially, there are computers, there are human knowledges, and desire to make the